Today we'll be looking at organelles, but before we look at specific organelles, we need to mention how there are two main types of cells. There are prokaryotic cells that contain no nucleus and eukaryotic cells that do contain nuclei. Let's start with prokaryotic cells. Prokaryotic cells are always unicellular. They have no need to form multicellular organisms because all their functions are contained within themselves. For example, this cell, influenza A, or this cell, Yersinia pestis, which you may know as the plague or the Black Death. Prokaryotic organisms are also more basic and tend to be older and more ancient than eukaryotic cells, and it's believed that eukaryotic cells evolved from prokaryotes. Prokaryotic cells also contain circular DNA found in nucleoids, which means like a nucleus, just like how humanoid means like a human while not being human. So prokaryotes don't have a nucleus, but they do have a nucleoid. This is what circular DNA looks like. Prokaryotes also have extrachromosomal DNA that float about in the cell named plasmids, and these are very good for research because you can easily take them out and add them back into these prokaryotes to study the effects the DNA being handled on the organism has. Most prokaryotes have cell walls and flagellum, or little tails for movement. Now for eukaryotic cells. Eukaryotic cells are found in multicellular organisms. However, not all eukaryotic cells are multicellular. For example, this is a euglanous cell, a eukaryotic unicellular organism. Eukaryotic cells also have membrane-bound organelles, including the nucleus. Prokaryotic cells do not have membrane-bound organelles. Prokaryotes are like 100 people all working in one space, where eukaryotes are the same way, except each person has their own room, with walls to work. It's far more productive to compartmentalize like this. Eukaryotes also have linear DNA, which is what we'd consider normal DNA. The first organelle we'll talk about is the nucleus, since we've mentioned it so many times already, whose main function is to store genetic material. We're starting with the nucleus because it's one of the easiest organelles to identify. This is a human cheek cell, and this is the nucleus. These are onion cells, and these two are the nuclei. Lastly, this is frog blood, and these are also the nuclei. This is a diagram of the nucleus. This is the nuclear envelope, which is a double membrane that separates the nucleus from the rest of the cell, demonstrating compartmentalization. This is a chromatin, or a mass of genetic material found in the nucleolus. This is where rRNA, or essentially subunits for ribosomal production, is made. These are nuclear pores, which allow for the selective passage of molecules in and out of the nucleus. This is the cisternae, which isn't part of the nucleus. It's part of the endoplasmic reticulum, but since they are right next to each other, the second organelle we'll look at is the endoplasmic reticulum, whose main function is to modify proteins and produce lipids. This is the endoplasmic reticulum, or the ER for short. It's a heavily folded membrane-enclosed space. The folding adds extra surface area to the ER, allowing for more space to perform the needed chemical reactions, once again showing compartmentalization. The space between the folds is called the lumen. Now there are two types of ER. The rough ER, which is dotted with protein-producing ribosomes, giving it the rough name, and the smooth ER, which has a more tubular look and whose function is to synthesize lipids or fats and to detoxify poisons and drugs. The AP Bio exam likes to make you apply your knowledge, so it's good to know what types of cells have more or less of a certain organelle. For example, plasma cells in blood often have a high ER count because they have to produce a lot of proteins, like antibodies, and rough ER's job is to produce proteins. Therefore, these plasma cells will have a higher ER count. Since we've already mentioned them, the third organelle we'll look at are ribosomes, which are found in all cells. Their main function is protein production. This is a ribosome, or a complex structure comprised of two subunits, one big, one small, and it's made of rRNA, or ribosomal RNA, which is made in the nucleolus, as well as other proteins. There are two types of ribosomes in cells: free ribosomes that live in the cytoplasm and produce proteins for the cell, and bound ribosomes, which are the ones we saw on the rough ER. Bound ribosomes produce proteins for exporting, like how plasma cells we mentioned earlier needed a high rough ER count, rough ER being dotted with bound ribosomes, to secrete antibodies for the body. This down here is the small unit, and this is the big unit. This chain you see is mRNA, and this green substance is tRNA, and the blue dots would be the amino acids being strung into a growing peptide chain, or essentially a protein. Let's look at the fourth organelle, the Golgi Apparatus, which can also be referred to as the Golgi body. The main function would be to modify, sort, and package proteins. This is a Golgi apparatus, or a stack of membrane-enclosed sacs located between the ER and the cellular membrane. Let's pretend that this side faces the ER. This side of the Golgi facing the ER is called the cis face. If the cell membrane is here, this side of the Golgi is called the trans face. This is the cisternae, or a flattened membrane disc. Most Golgi bodies have around 6 of these, however numbers can range from 20 to 3. Just like in the ER, the space between these membranes is called the lumen. This is an incoming transport vesicle coming from the ER. It gives its contents, which are likely proteins, to the Golgi, which modifies them and then packages them into an outgoing transport vesicle. These outgoing transport vesicles bubble off the transface towards the cell membrane for exporting. Remember, the ER's ribosomes produce material for outside of the cell. The fifth organelle we'll look at are lysosomes, whose main function is cellular digestion. This is a cell, not a lysosome, which would be a membrane-bound sac of hydrolytic enzymes that acts as a mini stomach. A lysosome can break down old organelles to recycle their parts, eat bacteria, digest food, and perform apoptosis, or programmed cell death, by releasing the enzymes within it to kill the cell if needed. This is the lysosome, and as you can see, unwanted foreign bodies, damaged organelles, and other unwanted molecules are brought to it. White blood cells tend to have a high lysosome count, since their function is to eat and kill off bacteria. The enzymes inside these lysosomes help break down and digest the pathogens so they can't further harm the body. The sixth organelle we'll look at is the mitochondria, whose function is to provide energy for the cell. This is a mitochondria, or a highly folded and double-membraned organelle, where cellular respiration takes place, or where ATP, or energy, is produced. This is the outer membrane, and this is the inner membrane, and the space in between is called the intermembrane space. These folds here are called the cristae, and the folding, like in the ER and Golgi body, increase surface area, and therefore the organelle's overall productivity. The big space in the mitochondria is called the mitochondrial matrix. This is also where the mitochondria stores its own DNA, and this can be something that can be used as evidence for the endosymbiont theory, which is a theory that organelles such as the mitochondria that have their own DNA were once freestanding prokaryotes that got engulfed by other prokaryotes, and this is how organelles or membrane-bound organelles came to be. Muscle cells are examples of cells that have a high mitochondrial count, as your mitochondria can produce all the energy your muscle cells need to keep you moving. Organelle number 7 is the peroxisome, whose main function is to break down fats. This is a peroxisome, named this way because it produces hydrogen peroxide which breaks down fats for energy. Peroxisomes also serve to detoxify poisons. It's comprised of a lipid bilayer which keeps the hydrogen peroxide from degrading the whole cell, and this is the crystalline core. The specifics of this organelle aren't that important to the exam, so long as you know that it can break down fats. Liver cells are examples of cells with a high peroxisome count, because peroxisomes can detoxify substances like alcohol, and it's the liver's job to keep your blood clean. Organelle number 8 would be the flagella and the cilia, whose main function is movement. Flagellum are very complex structures, and you won't need to know the details of how one is built and how one works, but it's good to know that they are little tails made from microtubules that rotate and allow for movement. Examples of cells with flagellum would be sperm cells, which need the tails to swim and move around. The ninth organelle would be chloroplasts, whose main function is to perform photosynthesis. This is what a chloroplast, or the double-membraned organelle that turns sunlight and water and carbon dioxide into energy, looks like. You'll need to know some of the main features of the chloroplast for the AP exam. This is the outer membrane, and this is the inner membrane. And like the mitochondria, the space in between is called the intermembrane space. The little discs here are called thylakoids, and a stack of thylakoids is called a grana. The inside of these thylakoids is called the lumen, and on the outside is a liquid called the stroma. In between the thylakoid sacs are bridges called the stroma lamellae, and they act as a skeleton for the chloroplasts, keeping the stacks in place. Essentially, the thylakoids are pancakes stacked up into a grana, and the stroma is just the thylakoid syrup. Organelle number 10 would be vacuoles, whose main function is for storage and structure. In plants and animals, vacuoles serve different core purposes. This is a plant cell, and the big blue unit inside is the central vacuole, and its membrane, the tonoplast. Animal cells do not have a central vacuole because in plants it's needed for structural pressure as they don't have bones like animals typically do. It also serves as a storage component for waste products. This is an animal cell. Here you can see the food particles entering the cell via endocytosis, where the particle presses into the cell membrane and then breaking off a part of the cell membrane into its own bubble. This broken off piece of cell membrane is now a food vacuole, which can work with a lysosome to form a digestive vacuole and digest food particles. Waste products from digestion are excreted through exocytosis, where the vacuole's membrane merges back with the cell membrane and spits out the junk. So in animals, instead of there being vacuoles for structural pressure, the vacuoles facilitate endo and exocytosis, or cellular eating and pooping. Organelle number 11 would be the cell membrane, which is also called the plasma membrane, or the cell's phospholipid bilayer, and its main function is to help the cell maintain an inner chemistry and keep the cell's contents in the cell, as well as be selectively permeable to the materials the cell needs and allow for the release of the things the cell does not. All cells have a cell membrane. It's often represented with a fluid mosaic model, which looks like this. This is the bilayer we mentioned, and it has two layers of phospholipids facing different directions. These are glycoproteins, or proteins with sugars attached on them that help cells recognize other cells, and also help to bind to any molecule the cell needs to be attached to. These are glycolipids, which are lipids with sugars on them, and their job is to facilitate cell-to-cell recognition and stabilize the membrane. This is a protein channel, and these help to bring large materials in and out of the cell. This is a cholesterol, which helps regulate the fluidity of the membrane, meaning it essentially stops the membrane, which is a fat, from melting in heat like a butter would in a pan or freezing in the cold. These are cytoskeletal filaments, which help give the cells structure and shape. These are integral membrane proteins, which serve as transporters, channels, receptors, and other things as well, and they are permanently embedded into the cell membrane. These are peripheral membrane proteins, which are not permanently adhered to the membrane. Instead, they are loosely bound to the surface. These play a role in signaling and maintaining cell shape and structure. Now, integral proteins have hydrophobic and hydrophilic areas, or water-fearing and water-loving areas, which is why they can be embedded throughout the membrane. The membrane also has both hydrophobic areas, like the tails of the phospholipid, and hydrophilic areas, like the head of the phospholipid. The hydrophobic and the hydrophobic areas attract, and the same goes for the hydrophilic regions. Peripheral proteins tend to be hydrophilic, so they don't get sucked through the hydrophobic heads of the membrane, and therefore don't have the same hydrophobic and hydrophilic properties that an integral membrane would have. The 12th organelle we'll look at is the cytoplasm, which is another organelle found in all cells. Its main function is for cell structure. So this is a cell, and here you can see the nucleus, the mitochondria, and the endoplasmic reticulum, and the Golgi, but the substance keeping all these suspended and holding everything in place is the cytoplasm. The cytoplasm is a thick solution made of mainly water, but in it you can still find things like salts and proteins. Again, this is found in all living things, and it keeps everything where it needs to be. To quickly summarize, prokaryotic cells have no nucleus, circular DNA, and no membrane-bound organelles. However, eukaryotic cells do. Eukaryotic cells also have linear DNA. All cells have a plasma membrane, ribosomes, and a cytoplasm, which can be used as proof that all life evolved from a common ancestor, since we all have these conserved traits. Lastly, the more surface area present, the more productive a cell is. The heavy folding seen in so many organelles like the mitochondria and the endoplasmic reticulum increases the space for chemical reactions to occur, and therefore increases cellular productivity. We hope you found this useful, and thank you for watching.